Yes, good morning, children. We are going with the principles of inheritance and variations. So, before proceeding with the concept, let us understand some of the important terminologies what we have read in our previous session. That is the genetics. It is the study of the scientific study of the inheritance and the variation is called as genetics. Okay, it is the branch of science that deals with the study of heredity and variation. Okay, now, what is heredity? It is the transmission of characters from the parents to offsprings. Okay, offsprings here means children, from the parent to children. If the characters are getting transferred, means that is what we'll call as heredity. Will be similar to our parents in our looks. Mostly will be similar. That is what the heredity. Then variation. It is the difference among the offsprings and with their parents. We are similar, at the same time there are differences also, there are some differences. Okay, that is what the variation is. And again, variation is important for evolution to adjust with the environment. Again, variations are important. Variation plays a very vital role in our survival, and they are the differences among the offsprings and with their parents. Okay, the next one is the hereditary variations. These are genetical and inheritable. Hereditary variations, these are genetical and inheritable. They are genetical and they will get inherited. Okay, means they will get transferred. These variations will get transferred from the parents to the offspring. That is what the hereditary variations are. For the best example, we can go here with this is the eye color. See, commonly will be having the black colored eye, otherwise the then brown, and the rarest one is the blue eyes, or the green eyes is the rarest of the rare one. So the difference in the eye color or the difference in the iris, which is responsible which is responsible for the eye color, is nothing but the hereditary variations. See, we all are humans. We all are having two eyes, one nose, one mouth, two hands, legs, and all. Yes, these are the similarities. But in few cases, hereditary variations will be there, which will get transferred. If the mother is having the brown eye, means may get transferred to her children. If the father is having blue eyes, means will get transferred, may get transferred to the children. That is what the hereditary variations are. The next one is the environmental variations. These are the acquired and inheritable. They are acquired, they are gained. Okay, they'll get gained, they'll not get transferred from the parents to the offsprings. That is what the environmental variations are. They are acquired and non-heritable, will not get transferred. The best example we can go with this is the obesity. Depends upon the eating habits. Depends upon our eating habits. The obesity we can go with. Yes, and here see the environmental variations. If we are going with obesity, can be generally can be detect easily. So these environmental variations are easily detectable also among the siblings only. Okay, so the best example here we can go is the obesity. Others are also there. For if smoking is there, means again the variations will be there. Okay, the problems will be there. That is what the variations are. Okay, so about all these things we have read in our previous session. Okay, so next we'll go with the experiments related with the principles of inheritance and the variations. See, this Gregor John Mendel is regarded as the father of modern genetics. Okay, he was the one who first elucidated the role of genes in transferring the characters. He was the one who first elucidated. So before him, many other scientists were working on this principle. Okay, so see, scientists like Skliden and Virchow and Squan. So these three scientists, they were working on the cell. They worked on the cell. Okay, they worked on the cell, and they elucidated that the cell will undergo binary fission, and from a single cell two parent cell will get formed. This is what the scientists said. Okay, they said that a single cell will grow in its size, it will grow in its cellular components, and then eventually the cell will get divided into two. This is what the Schleiden, Virchow and Squine has said. Okay, and they also elucidated the mitosis as well as meiosis. They also read about the mitosis which will be happening in the somatic cells, and they also studied about the meiosis which will be happening in the gametic cells around 1800 itself, before the Mendel. Before Mendel itself, they read about the cell. Okay, and they also deduced the link between the parent cell and the daughter cells. That is, they also said that the link between the parent cell and the daughter cell is the nuclear part. They said that nuclear, the nuclear component will get transferred from the parents to the offsprings. And they said that nuclear part, that is the nucleus, okay, that is the nucleus is the link between the parent and the daughter cells, has been said by these three scientists itself. But they fail to elaborate, but uh, they fail to hypothesize the transfer of characters from the parents to the offsprings. They said that the link between the parent and the daughter cell is the nucleus. They said this, that is correct only. It is the nucleus. Within the nucleus only our DNA is there. Within that the genes are there. They were correct only. They were true only. But they failed to explain it. They didn't explain that that is the nucleus which is responsible for the transfer of characters from the parent to the daughter cells. And this has been elucidated by the Mendel. And that is the reason he is known as the father of modern genetics. Okay, the experiment started around 1800 itself. At the starting of the 19th century itself, the experiment got started and has been started by the Schleiden, Squine, and Virchow. Okay, these three scientists, they studied the cell, and they elucidated that the cell will grow in size and eventually the cell will divide. That is what we read in our cell division. First, the cell will grow in its size. That is what we'll call it as the interphase or the growth phase. The cell will grow in its size. The cell will attain a particular size, and then after attaining that particular size, the cell will undergo the cell division. Okay, the same thing they too said, and they even elucidated the mitosis and the meiosis. Mitosis will be there in the somatic cells, and meiosis will be there in the gametic cells. They also said that there will be a link between the parent and the daughter cell, and that link is the nucleus. The nuclear compartment, the nuclear part is the link between the parent and the daughter cell. They also elucidated this. But they fail to explain the transfer of characters will be there by means of this nuclear part. They fail to describe this. Okay, and that has been elucidated by Mendel. Okay, Gregor John Mendel, he was the one who actually elucidated the transfer of characters from the parents to the offsprings, and that is why he has been regarded as the father of modern genetics. And he has been regarded as the father of modern genetics after his death. After around 16 years of his death, he has been regarded as the father of genetics. That is what, we should keep doing our work. If we are correct, means we will get the fruit one or the other time in our life. We will get it. That is what happened with him. He has been regarded as the father of genetics. Okay, so Gregor Mendel developed the principles of heredity. Okay, and he was the one he was the one who actually applied statistical analysis and mathematical logic for the biology. Okay, so Gregor is the Mendel of modern genetics, and he conducted the hybridization experiments. We'll be reading about that in this as the father of genetics, and he conducted the hybridization experiment for around 7 years. Just see the time. 7 years continuously he worked. Okay, that is between 1856 to 1863 he worked and proposed the law of inheritance. Okay, and for this he applied statistical analysis and mathematical logic. Because of these two reasons his theory got rejected. Because of these two, as he applied statistical analysis and mathematics to the biology, most of the scientists over that period of time they rejected his work. Okay, and his work went unnoticed because of these two reasons. But he was correct what he was doing. For his 7 years of his hard work, okay, after that much of hard work, he postulated his work in the form of statistical analysis and mathematics. And as he's applying statistics and mathematics in biology, his work got rejected. Okay, so now, what actually he did in his experiments? He actually conducted hybridization experiment. Okay, hybridization experiment he conducted. Now, what is hybridization? Hybridization is artificial pollination or cross pollination. Hybridization means development of a plant breed by means of artificial pollination or by cross pollination. That is what we'll call it as hybridization. Forming a plant breed, a improved plant breed or a plant breed by means of artificial pollination or by means of cross pollination is called as hybridization. For how many years he did? He conducted these experiments for around 7 years. He conducted his experiments and then he gave his theory. And we are talking here about the pollination, artificial pollination and cross pollination. Pollination will be there in the plants. See, hybridization can be done with any organism. He might have went with animals also, but the process will be too complicated over there. That is the reason he went with the garden pea plant. Okay, the biological name is Pisum. Scientific name is Pisum sativum. Okay, on which plant he did his experiments? He did his experiments on the garden pea plant, that is the Pisum sativum. See, again, see, many different plants are there. Many are there. Then why exactly went with the Pisum sativum only? Why? What might be the reason? The reason is cultivating Pisum sativum is very easy. Okay, growing Pisum sativum is very easy. One reason is that the pure varieties are available. Okay, pure varieties means same set of genes are available. Okay, I'll be explaining about that. Then pea plants are easy to cultivate. They're easy to cultivate. Okay, as they're easy to cultivate because the life cycle of the plants will have only few months. Okay, the life cycle will be having only few months, means the life cycle will get completed in one season itself. In a single season, the life cycle will get completed in one season. In one season the life cycle will get completed. What is this life cycle? That is zygote to seed. Okay, seed germination will be there. Seed germination, afterwards vegetative phase will be there, right? You have read in your botany. After vegetative phase, then the reproductive phase will be there. Yes, after vegetative phase, reproductive phase will be there. This is what the life cycle. So this complete life cycle will get completed in one season. And after reproductive phase, after producing the peas, the plant will die. This is important. The plant should have a smaller life cycle so that the observation will be easy. In around 3 to 4 months, this plant will complete its life cycle. If the Mendel might have been went with this neem plant 100 years, mango plant 60 to 70 years, can he wait? One plant will take so many years. And again, killing should not be there. A plant should the plant should have a smaller life cycle. So one reason is that also, that as this pea plant is having a smaller life cycle, there is only few months, he went with the garden pea plant. At the same time, plants are easily available. Okay, they are easy to cultivate. Just in the garden we can cultivate. That is the reason he went. Further, see, he went with the garden pea plants because of the contrasting traits. Okay, contrasting traits means for every character there will be two traits. For example, if we are going with the length of this plant, if we are going with the length of this plant means here, okay, if you are going with the length of this plant means, so length is the character, length is the character. Okay, so the contrasting trait will be the plant will be either tall, the plant will be either dwarf. So these are the two contrasting traits. These plant exhibits contrasting traits, and these contrasting traits are easily easily can be observed. That is the reason again he went with this plant. Okay, contrasting traits are observed easily, we can just see them. Okay, no need of any microscope, no need of any chemical experiment or any chemical reaction to see these contrasting traits. These traits will get expressed in the plant, and easily we can observe it. And that is the reason again he chose this garden pea plant. Okay, further, another important reason is flowers are bisexual and normally self pollinated. Okay, bisexual flowers here in the sense bisexual, both the both the male and female gamete will be there in a single flower. That is what here, the flowers are bisexual. This is important to get the pure lines. Okay, this is important to get the pure lines. That is the reason he went with the pea plant. Another one, flowers can be cross pollinated only manually. Flowers can be cross pollinated. We can cross pollinate the flowers. And again, that is the reason he went. And further, very important, hybrids are fertile. See, he continued his experiment for around 7 years. For 7 years, he worked with the garden pea plant. Yes, in the sense, if he has worked on that, means it is important for this plant to be fertile as hybrids. If the hybrids are fertile, means then only he can continue, otherwise he can't continue. So in this case, in pea plants, the hybrids will be fertile. That is the reason he went with the garden pea plant. Another important reason is these flowers, the flowers of these plants will get closed very tightly. They'll get closed very tightly, and because of which again the pollen grains will not leave the flower, and the pollen grains can't enter into the flower, which again aid in the generation of pure lines. The flowers of these plants will get closed very very tightly, so that the pollen grains can't enter from outside, and the pollen grains which are there in the flowers will not leave the flower, because of which the pure lines can be produced, the self pollination can be done. Because of that reason again he went with this plant. This is one of the important part in this chapter with the theory point of view, that why Mendel selected only pea plant. The reason is again, pure varieties are available, pea plants are easy to cultivate, life cycle of plants are only few months. In one season, the life cycle will get completed and the plant will die producing the seeds. That is the reason. Contrasting traits are there. Okay, so this one we'll be reading now. So contrasting traits. So contrasting traits means one character will be having two traits, that one. Flowers are bisexual. This is important again to get the pure lines. To get the pure lines. Okay, it is important, and normally self-pollinated. Easily they'll self-pollinate themselves. Flowers can be cross-pollinated. Okay, and then hybrids are fertile. Further, the flowers will get closed very tightly, and because of this reason all these reasons, Mendel went with the garden pea plant. Okay, this is one of the very important part with theory point of view, as again one of the most important chapter this is. Okay, so this is why he went with the plants, garden pea plants. Okay, further see, in his experiment, Mendel took around 14 plants. Okay, how many plants Mendel took? Mendel took around 14 plants. Mendel took around 14 plants. Why he took around 14 plants? Because he chose 7 characters. He took 7 noticeable characters, easily observable characters. So Mendel took around 14 plants. Why 14? Because he took 7 characters which are easily observable, which can be seen easily. Why he took 14 means each character will be having 2 traits. That is what the contrasting characters, what we read just now. Each character will be having 2 traits. 7 twos are 14. That is the reason. That is the reason he, okay, he took 4 plants. So actually 14 traits will be there. So each plant with 1 trait. And again see, at a time Mendel read 7 characters are height of the plant, seed shape, seed color, seed coat color, pod shape, pod color, flower position. These are the 7 characters what Mendel chose in his experiments. These are the 7 characters. What are the characters? Height, seed shape, seed color, seed coat color, pod shape, pod color, flower positions. These are the 7 characters what Mendel chose. Now each character will be having 2 contrasting traits. Each character, height, for example, height is having 2 contrasting traits. That is, either the plant will be tall or the plant will be short. These are the 2 contrasting traits what the plant will be having. The plant may be either tall or the plant may be either dwarf. Okay, that is what, this is the character, and these are the 2 contrasting traits. Okay, so what are the traits? They are the contrasting traits, and they are also called as, they are also contrasting traits, and they are also called as allelomorphic traits. They are also called as allelomorphic traits. One character, 2 traits. So for height, either the plant will be tall or the plant will be dwarf. No, both. It can't be tall and dwarf at the same time. Okay, it can't be. So either it will be tall or it may be dwarf if it is going with the height. In case of seed shape, either it will be round or will be wrinkled. Okay, seed color, seed may be yellow in color or might be green in color. Seed coat color, if we are taking, the seed coat might be green in color or it may be yellow in color. Pod shape, either the inflated, or constricted. So these are the constrictions. Okay, so either a softness will be there or the constriction will be there. So this is about the pod shape with 2 contrasting characters. Pod color, the pod color may be green, it may be yellow. And then the flower position, either the placement will be axial or the placement will be terminal. This is what the character, the flower position, this is the character, and these are the 2 traits. So these 7 characters the Mendel chose these 7 characters. Okay, these 7 characters he chose, and as these characters are having 2 traits, he took 14 plants of that particular trait. And at a time, Mendel read only one plant, only one character at a time. At a time, he didn't went with all the 7 characters. If he might have done means it might be very, very complicated. He can't. It will be very complex. That is the reason at a time he went with only one character. Okay, so how he did means, so he took the 14 plants and then he allowed the plants to self undergo self fertilization. What he did, he took the plant and he made the plant to undergo self fertilization. Okay, now what is self fertilization? Self fertilization means fertilizing within themselves. As these flowers are bisexual, as these flowers are bisexual, easily they underwent self fertilization. So bisexual means both the male and the female gamete will be there in the same flower. Both the male and the female gamete will be there in the same flower, and he allowed them. And then again, see, multiple self fertilization he did. He allowed the plant to undergo multiple self fertilization. So first self fertilization, he'll get a seed from, and again he has sown this seed. Again the plant came. Again seed from, again the seed came. Again he allowed that seed to undergo to germinate. Again he has sown the seeds. Again the plant germinated. Again self fertilization he allowed. Like this he kept on doing multiple self fertilization, and at last he got the pure lines. Okay, what he got? He got the pure lines. So pure lines are the plants which will will give the same character after multiple fertilization, self fertilization. Okay, those are the pure lines. If we are allowing these pure lines to self fertilize, they will give the same characters for multiple divisions. For example, if the plant is tall, if the pure plant is tall means, if we are allowing this plant to undergo self fertilization means, the coming progeny will be tall only. The coming progeny won't be short at all. Okay, the coming all the progenies will be tall. That is what the pure lines. Pure lines means these plants will be having the genes only for either of the trait. Pure, no mixing will be there. Either of the trait, either the tall trait or the round one or the wrinkled one, anyone, but only those genes will be there. That is what the pure lines are. So first he allowed the plant to undergo self fertilization, and afterwards he cross pollinated the plant. So first is self pollination, afterwards he cross pollinated. Okay, so how he cross pollinated? He took the flower, he removed the anthers from the flower. What he did, he took the flower and he removed the anthers with the help of a scissor. He just cut and removed the anthers, and that process is called as emasculation. What is emasculation? Removal of anthers. Removal of anthers from the flower is called as emasculation. Okay, so for cross fertilization, he took the flower, he removed the anthers with the help of a scissor, and that is called as emasculation. These anther he transferred to another flower of other plant, other emasculated plant. Got it? In that way, he did the cross pollinated and he formulated the theories. Okay, so and see, after doing all this, he had a keen observation. For around 7 years, he did these experiments and he had a keen observation. He recorded the data, he hypothesized his data, he did the multiple experiments on his hypothesis, and then what result he got, he took and he tabulated by means of statistics, and he applied the mathematics also here. Okay, that we will be reading in our coming sessions. So he did all these things and he formulated the postulates, and he said that the transfer of characters, the hereditary characters, will be there from the parent to the offsprings by means of genes. And to these genes, he called these genes as Mendel's factors. And he, by doing all these experiments, he elucidated that the characters will get transferred from the parents to the offsprings by means of genes. And what he called to those genes, he called the genes as Mendel's factor. And then he formulated his postulates, and it was Correns. Okay, see, before Correns, and he formulated his postulates. These postulates got rejected by many of the scientists in that late 1800. Many of the scientists rejected his theory, rejected his hypothesis. That too, this hypothesis was there with the clear cut results. Okay, there was no mixing and all, was there was no manipulation at all. But that too, the hypothesis got rejected by many of the scientists because he had applied statistics and mathematics for the first time in biology. As he has applied statistics and especially mathematics first time in biology, his theory got rejected. That is what, the tug between the mathematics and biology. Okay, that is what, see, in those then that time also, the biology lovers hated mathematics, right? That is what the connection, it is again, see, it is again from many years, it has got heredited. Okay, it has got transferred, because as he used mathematics, it got rejected. Okay, meanwhile he died, and after around 16 years of his hard work, after around 16 years of his death, okay, it was Correns who actually proposed Mendel's postulates as Mendel's as Mendel's laws of inheritance. Okay, Mendel's laws of inheritance. It was Correns. After his death, it was Correns who again rechecked his hypothesis by himself. He did the experiment, and he published, okay, the postulates of the Mendel as the Mendel's law of inheritance. And then he has been regarded as the father of genetics. Okay, poor fellow, he even don't know that he is the father of modern genetics, right? He even don't know that he is the father of modern genetics. He just did his work. As his theory got rejected, he just kept quiet. He didn't did anything, he just left it. Okay, because everything what he has done, he has noted. But see, after his death, again the checking did by the Correns, and again, the again the he just postulated it as the Mendel's law of inheritance. Okay, now what does the law of inheritance includes? Law of inheritance includes two important, very important laws. That is, first is the law of segregation. First is the law of segregation, and the another one is the law of law of independent law of independent assortment. Okay, law of segregation and law of independent assortment, these are the two important laws from the Mendel's law of inheritance. One more important postulate is there. It is called as law of dominance. It is also very important actually, but many problems are there with this. Okay, law of dominance. Okay, whereas these two are important, even this is also important, but we will be reading in our coming sessions. Okay, so law of segregation and law of independent assortment again has been given by Mendel, and law of dominance is also given by Mendel, and that is the reason he has been regarded as the father of genetics. Okay, so in our coming session, we will be going with the inheritance of one gene. For time being now, I'll be ending my session. Thank you.